We're going to examine this question at two different levels. We've got our original DNA sequence, and this is the sense strand of the DNA, so it looks just like the RNA sequence, except we're got to put U's in wherever there's a T. So this is the strand that matches our mRNA code. And we're asking, what happens if you mutate this sequence? We're going to mutate it in different ways. You can see AUG is our start codon. That's the methionine, so that's where all the machinery will start making the protein. Let's look at each of these, break each of these up into threes as well. And see what the effects of different mutations are. So with this first one, we've got just one spot that's different. That C there is different from the G, and everything else is the same. So we call this a point mutation. Look at the next one. On this next one, it's one shorter. So this last codon is missing a letter. And where does that happen? That happens with this T. There is a missing T. That is called a deletion. In the third one, we have, if you go along and check it closely, everything's the same to here. There's an extra C, and then it goes back to TCC just like at the top, AGT, AGT, TAG, TAG, but we've got an extra letter in the code, and that means we've got an extra letter there where the codon doesn't have all three. This is called an insertion. Now we want to decide if these are silent mutations or noisy mutations. It's not always one or the other. It's not always perfectly clear, but we can look at our sheet of RNA codons, and you take a look at yours or a chart that you find online, and we'll go through and label these. Our AUG gives us methionine, ACG, threonine, UUU, phenylalanine, UCC, serine, AGU is another serine, and UAG is a stop codon. So what happens if we just change one letter? Well, methionine's the same, and now we're looking at ACC instead of ACG, and ACC is also threonine. Phenylalanine, serine, serine, stop, all the same. So nothing has changed. This is a silent mutation. So many mutations don't cause any problems at all and don't even change proteins at all. Or Maybe they'll change one amino acid, but it won't be a problem. Let's look at the others. We start out the same. Methionine, threonine, phenylalanine. Now we go to CCA. Checking our chart gives us proline. GTT is GUU in the RNA, gives us valine. And AG is not a stop. The machinery won't know to stop. So this deletion changes the protein in significant ways, changes 2 amino acids out of the 5 here, changes everything from there on, and we lose the stop. So it's very noisy. It would make this gene non-functional in almost every case. Okay, this one we added a C. So let's look up CTC, that's CUC for RNA. CAG, glutamine, UUA, it's going to give us leucine, and G is again not stop, so the machinery will have a hard time knowing where to stop. So we went from serine, and if you look up the structures of these, serine has a hydrogen bonding alcohol group on it. So 2 serines with hydrogen bonding alcohol groups to a leucine, which is all alkyl side chains, all nonpolar. And we've got 2 of them, and it doesn't know when to stop. And glutamine, that can hydrogen bond, but it's bigger than serine. So big changes here. This is a very noisy mutation. So point mutations are often silent. Deletion and insertion mutations are almost always very noisy. So those are good choices to assume on your problems when you encounter a point mutation, a deletion, or an insertion mutation.